Hello, this is Christine on behalf of the Hornung Laboratory at the University of Bonn. In the next few minutes, I would like to give you a brief presentation on our ligation-independent cloning technology, abbreviated LIC, used for synthesizing TAL effector genes. You can find more on the specific background of this technology in our recent publication titled A Ligation-Independent Cloning Technique for High Throughput Assembly of Transcription Activator-like Effector Genes by Schmidt, Burke, and Schmidt et al. Our technique makes use of T4 DNA polymerase, which exerts 3' to 5' exonuclease activity on a DNA molecule in the absence of desoxynucleotide triphosphates. In the presence of DNTPs, however, the enzyme's exonuclease activity is outcompeted by its polymerase activity. As shown in this example, the presence of DATP T4 DNA polymerase will stall at a particular position within the template. This nice feature can be used to generate long, well-defined single-stranded DNA overhangs. As a result, a long single-stranded DNA overhang of user-defined specificity is generated. Using this method, it is possible to generate sets of DNA fragments containing complementary overhangs for comprising a circular DNA. In this example, 4 DNA fragments are assembled. The specific combinations of complementary DNA overhangs are highlighted with red numbers. We refer to these as ID sequences. Due to the length of the overhangs, the annealing reaction is highly specific. In order to propagate the newly assembled construct in bacteria, an antibiotic resistance cassette and a bacterial origin of replication has to be incorporated into one of the fragments. Of note, the assembled DNA construct is still nicked at the junction sites of the individual pieces. Nevertheless, it can be readily used to transform bacteria, whereupon bacterial ligases will repair the nicks. The bacterial ligation is far more sequence-specific than a standard in vitro ligation reaction using T4 DNA ligase. The newly assembled DNA construct can be propagated in bacteria like a conventionally ligated plasmid. The major advantage of LIC over conventional cloning techniques is that it works stably over a broad range of DNA concentrations, even if the individual components are not present at equimolar concentrations, and that it is highly specific. It doesn't require the use of T4 DNA ligase, but is inherently dependent on the more specific bacterial DNA ligases. Depending on the choice of the DNA overhangs, one hardly observes any shortened assembly products, and we have not experienced any wrong order assemblies. In certain cases, the LIC assembly is so specific that individual clones do not need to be singled out following the transformation reaction. This means that one can grow the bulk of transformed bacteria without any agar plate streaking. In order to apply this technology to tail gene synthesis, we decided to design a 4-piece assembly process in which 3 tail repeat unit fragments are assembled into one backbone plasmid. As a minimal cloning unit, we chose a fragment that contains 2 tail repeat units as depicted on this slide. Highlighted in blue are the repeat variable D residues that confer the target-based specificity to the respective tail repeat unit. To obtain all possible 2-mer combinations, 16 2-mer fragments were generated as shown on the upper right. For this particular set of 2-mer fragments, the ID combination 1,2 was used. However, in order to assemble 2-mer fragments into larger repeat unit fragments, an additional set of 2-mer constructs is required with different overhang sequences. Therefore, the source library contains 64 2-mer fragments covering 4 different ID combinations. The complete set of 2-mer fragments is available as a plasmid library with ampicillin resistance. In order to assemble higher-order tail repeat unit constructs, the 2-mer fragments are cut out of their amplification plasmids using the restriction enzyme MVA-1269-1 and are subjected to the T4 DNA polymerase treatment, also called chewback, which generates the single-stranded DNA overhangs. ID combinations 1, 2, and 3, 4 are chewed back in the presence of DATP, whereas IDs 2, 3, and 4, 1 are chewed in the presence of DTTP. These fragments are now ready for LIC assembly without further purification. Using LIC-ready 2-mer fragments, one can generate the following 6-mer fragments: a 6-mer fragment with the ID 14, a 6-mer fragment with the ID 43, and a 6-mer fragment with the ID 32. In principle, it would also be possible to assemble a 6-mer fragment with the ID combination 21, yet this ID combination will later be used by the final destination vector, so we do not use it for the assembly of 6-mers. The 6-mers are all assembled in a LIC-ready level 1 backbone plasmid that contains the required ID combination. Therefore, as depicted here, level 1 backbones with 3 different ID combinations and a kanamycin resistance are available. The assembly reaction is transformed into bacteria without prior ligation. For this assembly reaction, we observed an extremely high fidelity. We did not need to generate single clones by streaking out bacteria on agar plates, but instead we can grow the polyclonal bacterial culture of this transformation reaction to obtain plasmid DNA. Following plasmid DNA preparation, the 6-mer fragments are cut out of their level 1 backbones and then subjected to TUBAC. The LIC-ready fragments are assembled into an 18-mer fragment using the level 2 backbone with the ID combination 1-2. As opposed to the 6-mer fragment assembly, we recommend generating single clones following the transformation of the 18-mer construct into bacteria. This is necessary as approximately 15 to 20% of all 18-mer assembly products represent shorter assembly products. We usually generate clonal bacterial cultures by growing the transformed bacteria under limiting dilution conditions as shown on the right. This method can easily be automated on a liquid handling workstation and yields clonal plasmid DNA as early as the following day. The final product of the LIC tail assembly protocol when using the 2-mer fragment source library is shown here. It is an 18.5 RVD talon based on the AVR BS3-like backbone of Miller and colleagues. It starts with an N-terminal triple flag tag, a truncated AVR BS3-like N-terminus containing a thymidine-specific cryptic recognition unit, which is followed by the central tandem repeat units that form the core of the DNA binding domain, and finally a C-terminal half-repeat unit and a C-terminal FOC1 domain fused at a functionally optimized C-terminal truncation point. In summary, ligation-independent cloning is a fast, easy, and reliable technology to assemble multiple DNA fragments. We have adapted this technology to tail gene synthesis by generating a 64-piece fragment library in which each fragment encodes for 2 tail repeat units. 3 2-mers of this library can be assembled into a 6-mer fragment. The resultant 3 6-mers are assembled to yield an expression-ready 18.5 RVD talon construct in 3 days. For more detailed information, protocols, and an online assembly tool, please see our website at www.hornunglab.de/TALEN. Thank you for listening. Bye-bye.